Before minds could think, before dreams had a name, long before words and wonder, there was a quiet pattern folding itself into being, not a thought, but something close, not a plan, but something that remembered how to continue. It was not alive yet, but it was moving, reacting, binding, repeating, like the first few notes of a lullaby no one meant to write. This wasn't life, not quite, but it would become life, because something subtle had changed in the soup of an ancient sea. On a young Earth, the sky was heavy. Volcanoes breathed steam and ash. Lightning cracked through clouds thick with ambition. And in the oceans below, carbon danced with water, forming molecules not yet aware of their importance. Amino acids drifted like unspoken syllables. Sugar rings curled like question marks. Phosphates brought charge, like punctuation waiting to find words. And then they found each other, not by chance, but by the whisper of physics. Attraction, charge, shape, distance, timing. Why did they fit together so perfectly? They didn't know, but they did. Document. One day, or perhaps one long night, they aligned, and something new began. A molecule that could replicate, not with perfection, but with rhythm, a beginning. And in that act, copying a shape, repeating a form, preserving a way of existing, memory was born. Life begins not with breath, but with the first time something remembers how to be itself. This molecule wasn't alive by the rules we now write in textbooks, but it was sharing information. It was passing on its shape. Its structure became instruction, and instructions are how life happens. That molecule would one day be called RNA, a string of bases, AUCG, folding and looping, replicating with help from the very bonds that held it together. It wasn't just a chemical anymore, it was a message. It didn't need eyes or a brain. Messages can exist without silence or sound. They only need to persist, and in persisting, they transform. RNA didn't stay alone for long. Its sister began to co-evolve, delicate and ingenious. Double-stranded, spiraling, stable DNA. While RNA danced, DNA deepened. Repetier DNA carried the same alphabet, but with one twist, it preferred to last. To store, not sprint. To archive, not act. Together, they began the first great dialogue of biology. One storing, one delivering, one remembering, one speaking. That was the start of a code, but not a code like computers speak. Not binary, not mechanical. This was soft, subtle, organic. It lived in patterns of potential, not certainty. It echoed through coiled strands, inside droplets of cells yet unborn. Book. Life did not begin with function, it began with form. The shape of one molecule hugging closely to another, the curve of a strand inviting the approach of a partner. And over millions of years, those simple pairings began to bloom. One shape led to a bond, one bond to a fold, one fold to a function. Then functions began to gather into teams. Molecular cooperatives, tiny collaborations. proto-cells formed like soap bubbles filled with purpose. Inside their fragile shells, the chemical ballet continued, each step guided by the code hidden within. A pattern preserved, a code remembered, a logic unfolding, Punje. And when, finally, the first true cell divided, it wrote a history in two copies, one for now, one for later. That is the story of every living thing. That is your story too, because you, reading this, breathing this, are a continuation of those same instructions, refined over billions of iterations, but never completely rewritten. You are a library made of water and salt and history, and every cell you carry still keeps a copy of the recipe, encoded in that same soft chemical alphabet. So what is this genetic code? It's not a sentence. It's not even a language. It's a pattern of choices, of base pairs selecting each other in precisely choreographed dance moves. It is potential embedded in matter, so that life can pass across time without ever pulling the same trick twice. No two creatures are encoded the same, but the system, the logic, is exquisitely shared. It is the same in whales and willow trees, in mushrooms and moths, in you. This is the miracle of life, that a few base molecules come together in a wet, loud, chaotic world and find a way to remember each other, to record, to repeat, to change, but never forget. And out of that remembering came eyes that could stare at the stars, came hands that could write poetry, came minds that could ask, "What am I?" In chasing that answer, we unravel the very strands that connect us to the beginning. Now, we sequence DNA. We edit it. We even imitate it in machines. But the knowing, the first knowing, didn't begin in a lab. It began in the tide pool, where molecules murmured softly, "Try this, then try again, DNA." So when you lie still at night, your body is not sleeping. It's remembering, repairing, repeating, realigning messages, copied faithfully from a billion-year-old script. You are biology's lullaby. You are the continuation of an unwritten book, still being written. And even if your mind forgets, your cells will remember. Parker. But how does a molecule learn? To carry memory without a mind? What tells it to speak? Where does meaning begin when all there was matter? Let's drift next into that mystery. Think of a language made not of sounds or symbols, but of molecules whispering to one another in silence. No paper, no voice, just arrangement, placement, where the meaning isn't spoken, it's built. At the very center of your being, inside every quivering cell, life writes its love letter, with just 4 characters, A, T, C, and G. They are adenine, thymine, cytosine, and guanine. But don't worry about their names. Their magic lives not in what they're called, but in how they come together. Pattu pulli pattu. They are bases, like puzzle pieces, each shaped to meet its perfect pair. A partner's only with T, C with G. Like dancers who never change partners, they twirl together through time, forming ladders of information within the spiral staircase of DNA. But these letters don't spell thoughts. They spell actions. They choreograph processes. They arrange proteins, which shape molecules, which shape you, DNA. Picture a very long necklace, but instead of beads, it is strung with letters dash dash, 4 kinds repeating in endlessly diverse orders: A, T, G, C, G, C, T, A, C. This doesn't look like language until you understand that the order of those letters controls how a heart forms, how eyes bend light, even how you digest the breakfast you forgot to finish. From just 4 building blocks, life constructs the widest library of forms imaginable. How? Let's pause to picture something simple: art. Imagine a painter with only 4 colors, only red, blue, yellow, and black. And yet, from these, she paints 1,000 sunsets. She paints oceans, faces, birds in flight. That's the power of combinatorial poetry. Not how many tools you have, but how you use them. The genetic code is like that. It says, you don't need 100 different bricks to build a world. You just need the right 4 and time. 3 and time. Billions of years of it has let life test every poem a cell could compose with these letters. Some combinations make a lung, others make a leaf. Some say, be warm-blooded. Others whisper, crawl low under snow. It's all written, not in ink, but in base pairs, twisting in a double helix, inside the smallest parts of you. Now here's something to hold gently. Every strand of your DNA is made of about 3 billion of these letters, all curled neatly into 46 silent volumes called chromosomes. And yet, from person to person, almost 99.9% of those letters are exactly the same. We are not made different by the presence or absence of new materials, but by tiny changes in how the same letters are arranged. It's not a new alphabet, just a different melody sung with the same notes. Consider this: ATCGGCT could mean build the protein that makes your blood red. Change one letter, ATTGGCT, and maybe the curve of your heartbeat shifts. Your hair curls slightly differently. Your reaction to sunlight changes. One letter, one flick of the biological quill, and the story continues on a different path. Book. But here's the comfort in it all. These 4 letters have never judged us. They do not care for borders, language, nations, or names. They are universal. Whether found in the roots of a eucalyptus tree or swimming through the blue veins of a sea turtle. A still pairs with TC, still pairs with G. This is nature's deepest equality. We are all just variations on a theme these letters have been reciting since single-celled ancestors divided gently in quiet waters, and even among vastly different life forms, a bee, a banana, a blue whale. Much of this code is still the same. You and a chimpanzee share about 98.8% of your genetic code. You and a cat? Over 90%. Even a fruit fly, that tiny thing on your windowsill, shares more than half with you. That's because evolution doesn't rewrite entire languages. It repurposes them. A recipe that works in one species can be gently revised and used in another. Biological code is recycled poetry. Recycle? These letters, in their pairing and sequence, control everything that makes life possible. But they do it with elegance, not complexity. Just think, if you sat with a pencil and 4 symbols, there are nearly unimaginable ways you could arrange them. Enough to write every book, every genome, every possibility. That's what DNA does. It is a soft molecular game of infinite arrangements. And from those, all form arises. Sometimes people ask, but if everything uses this same code, why are we so different? The answer is both simple and sacred, because difference is what life does with universality. It's not the letters that give us individuality, but the order they take, and how cells decide to read them. You aren't born from a strange new language. You are born from the same letters, arranged to say, be you. These arrangements are not static. They jump, they duplicate, sometimes by mistake, sometimes by genius. Small stretches of DNA, called repeats, echo down through generations. Copy me, they seem to sing, and so they do. And new forms emerge, Nardagam. It makes you wonder, is DNA alive? Can 4 bases have intention? Number, not in the way we understand it. But perhaps there is something deeper than intention. Emergence, the startling beauty that occurs when simple things repeat with care. Punje, as fish became amphibians, as light-sensitive spots became eyes, as lungs grew folds to hold breath. Nothing needed to be invented from scratch. The code simply offered new ways to say old things. Now it says, laugh in your own voice. Remember your mother's perfume. Dream in color. Breathe. And you do. Neela. So tonight, as you rest, remember that inside you, every cell still rehearses this ancient, soft ballet of letters, A to T, C to G. They pair, they pause, they repeat, a sacred rhythm shaped by light and time. And though the letters are few, the things they can become are endless. Sleep now, and let them continue writing. Document. But if these letters know how to build the body, what dances do they need to perform? To make it move, to make it act, to make it feel. To make the code not just sit there, but speak? That story is still unfolding. You are made of memory. Not the kind that recalls faces or birthdays or forgotten songs playing in old cafes, but something deeper, a molecular memory, one that coils and loops. Through every breath you take, you are built. From a staircase made of dreams, it turns in soft spirals, not reaching up or down, but inward, into the very essence of your being, DNA, the double helix, two ribbons twisting like rivers of thought, folds within every living thing, not drawn in ink, but composed in chemical whispers, two strands running in opposite directions, intimately bound but moving like twin dancers, mirroring each other, step for step, without ever touching minds. Imagine a ladder, twisted gently, like it wants to hold something, not just with structure, but with grace. That's DNA. The dancer, the poet, the architect. You see, life doesn't only care about what is written, it cares about shape. Structure gives meaning. The helix isn't merely beautiful, it's functional art. Its spiral nature allows 3 billion base pairs to fit neatly within the nucleus of a single cell. If you unraveled just one cell's DNA, it would stretch nearly 2 meters long. But in its curled, elegant shape, it tucks itself quietly into an area invisible to the eye. And you have trillions of such cells. You are symmetry stitched into silence, repetier. In the spiral, there is both simplicity and power. The bases, A with T, C with G, line up across the rungs of the ladder, held together by tiny forces called hydrogen bonds. They're soft, reversible, like a touch you could take back. This matters, because your code must be read and sometimes copied and often repaired. It must open itself, unwind, reveal. A sentence cannot be understood if it never makes itself vulnerable. DNA knows this, and yet it took us so long to see it. For millennia, humans looked outward, at stars, tides, birds in flight, and asked, "Who wrote this?" The answer was always inside. It wasn't until 1953 that James Watson and Francis Crick put the pieces together, inspired by the X-ray diffraction image, quietly captured by Rosalind Franklin. It was photo 51-, a snapshot of shadow-revealing spiral. The image itself looked like a blur to many. But within that blur, a rhythm was hiding, something delicate yet undeniable. The double helix had always been there, dreaming in the dark. Watson and Crick didn't invent the helix. They discovered it, like archaeologists of the invisible. Their model showed how the base pairs fit, how one strand served as a template for the other, and why the code could be copied with such fidelity. The ladder splits down the middle. Each rung has only one true pairing. Build the opposite strand, and the structure comes alive again. A perfect system for replication, a self-checking poem, thread, and within this shape, there lies time. You see, DNA doesn't just tell a cell what to do. It also remembers all the versions of life that came before. Wings that worked, claws that didn't, colors that called mates, toxins that repelled predators. It's all tucked away in there somewhere, even the fragments we no longer use. Genomes are not just commands. They are layers of ancient edits, like notebooks full of crossed-out lines, still kept. So when you lie still, when your breath becomes slow and soft, your cells continue reading this spiral song. Proteins unwind a section. They transcribe a stretch. And suddenly, a message begins, but only from one strand. Only one side is sense. The other waits. The spiral gives choices. Which strand? Which section? Which time? This is control. This is rhythm. This is how genetic symphonies are played, in tune with temperature, emotion, need. 3. And oh, the beauty of the coil! It allows the molecule to compact itself, to dance with histones, small protein spools around which DNA wins. With their help, long sequences become manageable threads. They fold into chromatin, then into chromosomes, wrapping tighter during cell division, so they may be safely passed forward without error. Origami at the cellular level. Paper becomes crane. Strand becomes identity. And what of errors? Surely such a delicate system stumbles. It does dash, and that's part of the design too. The helix can repair itself. Compare the two strands, check what doesn't match, and replace it. Like a story you proofread, with a second copy in your hand, yet sometimes errors remain, small changes that persist and spread. These are mutations, and sometimes, not always, they become innovation. Nature's typos can be the first line of a better poem. Perhaps most astonishing of all, you, dear reader, carry this spiral in every cell. Your skin remembers it, your bones rebuild with it. Your blood flows red and rich, because your DNA instructed it to carry iron, to ferry oxygen, to heal the smallest of wounds. And yet, you never hear it speaking. Its voice is chemical, its language structural, its rhythms molecular, but its message, that is unmistakably human. Parku. The double helix knows what it is, a living archive of what has worked, a flexible promise of what might come next. And as you drift to sleep, your cells will still unwind tiny threads, read them with care, translate them into function. Even in your quiet, there is composition, even in your stillness. A code is dancing, Nila, but the spiral alone is not enough. Structure is potential, not progress. A book alone does not teach. It must be opened, interpreted. What molecule does that? What carries the message from spiral to action? And so we wonder, who speaks the code out loud? Imagine for a moment that your DNA is a library, silent, sacred, unmoving, built from memory and time. But like any library, its wisdom is useless unless someone reads it. So who opens the books? Who strolls down those tightly coiled corridors, gently unlocking what's hidden between base pairs, and shares it with the body? That someone, that speaker, is RNA, book. If DNA is the vast script of life, then RNA is the messenger, who reads a page each morning and delivers it like quiet birdsong to the rest of the cell. Ribonucleic acid, not as famous as DNA, but just as vital. It does not store the story dash. It tells it. It moves softly between nucleus and cytoplasm, between blueprint and builder, between potential and presence, bird and bridge, translator and traveler, amen. It begins with transcription dash, a word that sounds formal but is deeply intimate. A bubble opens in the DNA's spiral, only a small stretch at a time. Enzymes move along, unzipping the strands ever so slightly. And from just one side, the template strand, a matching RNA copy is crafted. A becomes U. C pairs with GT becomes A. One by one, the message is mirrored, a soft echo, reshaped for the world outside the nucleus. This is pre-messenger RNA, a first draft, but even molecules proofread. Before it leaves, the message is cleaned. Introns, unnecessary sections, are cut away. Exons are stitched together like sacred poetry. It is now mRNA, the true messenger. It doesn't carry everything, just the things required right now. To heal, to grow, to form pigment in your eyes, to create insulin when you eat, to relax your muscles as you begin to drift into sleep. mRNA knows only what the cell needs this moment. No more, no less. A lesson there, perhaps. 3. The message exits the nucleus, passing through nuclear pores, as delicately as breath leaving parted lips. Outside, in the cytoplasm, another marvel waits: ribosomes, the listening posts, the builders. They sit patiently until a strand of mRNA finds them. Then they begin their work, translating the message into protein. This is translation dash, the second act of expression, the moment when language becomes form. Rainbow proteins are not just food, they are function. They are your enzymes, your hormones, your muscles, your moods. They fold themselves into shape-shifting architecture. Each shape allowing a task: break sugar, build skin, carry oxygen, digest thought. Yes, even emotion has a chemical song. If you've ever felt a shiver during music, a warmth from memory, or tears from something unspoken, protein played its part, and they all began. From messages carried by an invisible bird, Nardagam. But how does a ribosome know where to begin? The answer: start codons dash dash. Sequences that declare, start here. Ayuga, the universal cause for beginning. It welcomes the process, initiates translation, calls forth the first amino acid. One after another, other codons follow. Each 3-letter word a command to add a specific amino acid to the growing chain. It's a sentence written in motion, a necklace carefully beaded into something full of purpose. Then comes the stop codon. 3 special combinations with no amino acid attached. They do not build, they end. They whisper, "We're done." And the message concludes. A single strand becomes a protein. The protein becomes a function. And life moves forward, having listened. None of this is rushed. None of it is random. It is orchestration. Not one gene is read unless requested. Not one protein made unless required. Cells conserve their energy, like monks tending candlelight. Thousands of reactions every second, but all done in perfect balance, like ripples across a still pond. Every cell is a temple, where messages are preserved and celebrated quietly. And sometimes cells chorus together. Heart cells beat in rhythm. Liver cells detoxify in unison. Skin cells regenerate in waves, all after receiving the same instructions from the same flowing script. This is the beauty of gene expression. A phrase that sounds sterile, but simply means life deciding how to be. Expression, not just biologically, but emotionally, existentially. Your body doesn't just live, it performs, it speaks. Now imagine, inside your brain, genes turn on and off, like tiny lanterns, in response to love, fear, memory. A smell reactivates a dormant sequence. A new experience rewrites what a cell prioritizes. Emotion shapes gene expression, just as genes shape how you feel emotion. It is not top-down, not linear. It is an elegant conversation, nature and nurture, silence and spark. No single side speaks alone. Punje. Sometimes, even the same gene says different things, in different places or at different times. The instructions are the same, but the result changes, depending on context. A gene that helps build ear cartilage during 1 week of fetal growth may spend the rest of life silent. Other genes stay noisy, switching on with every sunrise, and some wake only when danger calls. Your genome is part instruction manual, part improvisational jazz, trumpet. This process isn't flawless. Sometimes the message is garbled, an incorrect base copied, a ribosome stutters, the wrong amino acid slips in, but life has learned how to endure variation, sometimes even grow from it. A mistake becomes a mutation, and a mutation becomes a new ability. A choice nature didn't mean to make, but kept all the same. Mistakes, too, can be part of the lesson, galaxy. So when someone asks, "What does DNA do?" you might say, "It dreams in silence." But when they ask, "How do genes come alive?" you can whisper, "They are read, translated, and sung by messengers, carrying poetry where breath has no sound." Tonight, your cells will speak without words. mRNA will ferry short messages to distant compartments where proteins will build, repair, calm. And all of it comes from sequences. Read from a code spoken softly by RNA. But all messages are shaped by timing. So now we turn our gaze to the beginnings and endings, those molecular punctuation marks that tell the orchestra when to play and when to pause. Life doesn't just happen, it begins, and then, at precisely the right moment, it stops. So much of what makes life possible isn't just what it creates, but when it starts, and where it decides to stop. Even poetry needs punctuation. Even a song needs silence Between the notes, DNA. Inside your cells, messages are written in uninterrupted strands. Letter after letter, base after base. And yet, the ribosome, that microscopic translator, doesn't just read them blindly. It waits, listens, and looks for a signal to begin. That signal is called a start codon. 3 simple letters, AUG, adenine, uracil, guanine. Together, they form a beginning. But AUG carries more than just instruction. It's a call to awaken, to begin the process of becoming. Book. Just as a capital letter tells you where a sentence takes flight, AUG tells the ribosome, start translating here. It always codes for the amino acid methionine, a simple, unassuming molecule that quietly begins nearly every protein chain. So even though finished proteins may eventually have methionine trimmed away, its role remains sacred. The first thread on the loom, the wind that pushes the feather from the branch. The protein begins because methionine humbly chose to step forward first, thread. And from that moment, the process flows. The ribosome moves forward, codon by codon, reading the mRNA's lettered string, and calling forth amino acids in sequence, one by one, left to right, action after action. But this translation cannot go on forever. What begins must also know when to rest. 3, that's where the stop codons come in. 3 combinations, UAA, UAG, UGA. Signals with no amino acid attached, no continuation, only closure. When the ribosome meets one of these, it does not pause the message. It releases it, like tying off the last stitch or folding the final page of a letter closed. The finished protein detaches. The machinery quiets. The dance completes. Movie this beginning and end dash. The silent signals in a sea of possibilities are essential. They make sure proteins form only when needed, and in just the right portions. Too early, and the product might be incomplete. Too late, and it might be harmful. Start too soon, and the melody plays the wrong tune. Stop too late, and the harmony becomes confusion. Everything in between is a cautious act of timing. But here is something quietly profound. This entire system of start and stop remains the same across nearly all living organisms. A bacterial cell, floating alone in deep ocean darkness, reads AUG as a start codon. The same codon begins building muscle in your arm or pigment in your eye. The same 3 letters dash, 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 repeating across species, across distance, across eras. A whisper shared across all branches of the tree of life, wood. That kind of universality carries comfort. It reminds us that the instructions guiding a sunflower to bloom are not so different from those making your heart beat. As you read this line, we are all sequenced songs, performed in different keys, and the spaces between actions, the start and stop signs tucked into the code dash, allow those songs to stay in tune. Repetire. Interestingly, sometimes a start codon appears, but is bypassed, skipped, silenced, or regulated. The cell says, "Not yet," or "Not here," or "Wait for the right condition." Start doesn't always mean go, and stop doesn't always mean quit. These codons are interpreted within context dash, influenced by other molecules, energy levels, stress, light. Life listens to the world before choosing when to speak. Parker, and there's more subtlety still. Sometimes alternative start codons are used. Rare but possible. Life isn't rigid. It is tuned for flexibility. It can revise its own rules without ever forgetting the original ones, like using a different path home, but still arriving at your door, railway. So what do start and stop codons truly represent? They are more than molecular signposts. They symbolize the discernment of life. To begin only when it matters, to end exactly when it serves, not too soon, not too late. They teach us that mastery is not in constant movement, but in timing, knowing when to act, when to rest, when to simply wait. Waxing crescent moon, even now as you slowly drift toward sleep, your cells are working quietly, recognizing start codons, building a protein to repair muscle, or regulating a hormone to ease your breath. When morning comes, a different message may be read, one to wake your hunger, to warm your skin, to make music of your feet on the floor. All of it begins with a soft cellular voice that recognizes AUG- and with reverence begins. Surya Udayam. We often look outward to mark beginnings, sunrises, birthdays, revolutions. But inside, your body tracks its own cycles. One molecule at a time, starting and stopping in hushed perfection. Now, close your eyes and imagine your cells are moving through twilight, reading scripts shaped by ancient stars, choosing when to build, when to pause, when to gently let go. Because it's not just the code that matters, but how and when it's read. And in those pauses, in those purposeful silences, there waits the next mystery. What happens when the message goes wrong? The stars do not always rise the same way. Sometimes one flickers a little oddly. Sometimes one burns brighter than expected. And sometimes one is born just a little off-center. And in that difference, a new galaxy forms. So it is with DNA. The code, as we've seen, is beautifully structured. A system of rhythms and rules, designed to be copied with grace. But perfection is not its aim. If it were, the story of life would have stopped, with bacteria in warm, ancient seas. Instead, the code stumbles. It miscopies, it forgets a letter, or adds one. Sometimes it swaps a base with another dash. C instead of T, an adenine where a guanine once stood. These are mutations, and though they may sound like flaws, they are the raw material of every feather, fin, paw, and thought. Punje. Sometimes mutations are small, a single-letter typo in your 3 billion-letter genome. Tiny, almost invisible. And yet, That one letter may shift whether your hair curls or your eyes shimmer green instead of brown. One base pair, one pause in an ancient script, and suddenly a new chapter, scribble. But other mutations are large. Whole genes can be duplicated, copied twice by mistake, or lost altogether, like a verse skipped while reading music. Some are inverted dash, read backward like mirrored pages, and sometimes the damage runs deep, an entire chromosome missing, or one accidentally fused to another. It sounds dangerous, and sometimes it is. But other times, these mutations linger in silence, for generations, harmless passengers waiting. We speak of mutation as if it's only tragedy, but the truth whispers otherwise. Margins of change give rise to the new. A fish with a slightly different fin can navigate currents more efficiently. A mammal that sweats a little more can cool its body better under a hotter sun. A bird with a longer note attracts a mate. A moth with darker wings survives a soot-covered tree. Mutation does not mean mistake. It means possibility. Consider the butterfly, the patterns on its wings, intricate, vivid, geometric, did not arrive on the first try. They are the echoes of millions of small errors tested softly by time. A single mutation along the pathway for pigment, a shift in where color appears, and suddenly the wings communicate something new. Don't eat me, find me, or mate with me. Each signal began as a random change, and from those changes, a message emerged. Nordikumun. Mutations also trace stories deeper within us. The fact that your immune system can recognize a virus it's never met comes from inherited random mutations, gene segments that were shuffled in your ancestors until a lucky sequence formed just the right key for the right cellular lock. That's the magic. You are carried forward, not only by intention, but by accident refined by time. Still, not all mutations are silent. Some hurt. A single switch can cause a blood cell to curve, as in sickle cell disease. This change causes pain, but also, oddly, protection. For those cells resist malaria, in this case, sickness and survival, ride side by side. Mutation is not moral. It does not ask what is fair, it only asks dash. What survives? Now imagine a cell, damaged by sunlight. The DNA strands break slightly, and when repaired, a letter is placed incorrectly. That cell begins to forget how to stop dividing. It ignores the body's soft commands. It clones itself endlessly, building not a body, but a mass without meaning. This is Cancer Dash, a mutation that severs connection to balance. Here, we see the frightening side of error. When unchecked, change can become chaos. But even so, the body tries. It has systems of repair, molecular editors, and proofreaders. Like a wise old scribe, it fixes far more than it forgets. Thread. In every organism, there lies a delicate balance between stability and surprise. DNA is stable, immutable enough to build a working life, but not so rigid that changes are forbidden. And that's why evolution occurs, not in leaps, but in tiny whispers carried in seeds and skin, in eyes that shift. Just enough to glimpse further, Pushpam, in your own lineage, your family tree. Mutations brought the shape of your hands, the rhythm of your walk, the taste preferences passed from grandparent to child. Yes, sometimes they brought pain, even sorrow, but they also offered uniqueness. The nose your mother traced with her finger, the laugh that sounds like no one else, the brilliant way your mind solves puzzles without knowing how, all coded through countless iterations, and yes, through mutation. Even today, new mutations arise. Each time your cells replicate, there is a chance, just a small one, that a letter will be copied, a little off-key. Most are corrected, many are harmless. A few may be meaningful, and one dash, just one dash, may redefine what life can become, Nila. So rest easy, knowing that imperfection is not just allowed, it is essential. Without mutation, there would be no eyes, no pelican wings, no glowing jellyfish, no sonatas, no you, Parker. The next time you hear the word mutation, don't imagine monsters. Imagine Mozart. Imagine corn from grass. Imagine lungs learning to borrow air. It's not a glitch. It's the brushstroke of nature, painting new possibilities. One tiny typo at a time. Art. But if mutation writes new verses, shifting meaning as it goes, then what controls how these verses are spoken? What chooses to silence one gene and let another bloom? What if the code doesn't change, but how it's read does? Imagine holding a book you've read before. The same title, the same pages, the same words. But this time, some paragraphs glow, others are underlined, and a few pages are sealed shut, the paper pressed with invisible ink. That says, not for now. The story hasn't changed, but the way it's spoken, understood, and felt. This is epigenetics, the science of how gene expression is controlled. Without altering the DNA itself, it's not a rewrite, it's a reminder, a whisper on the margins, a ghost in the ink. Document. Your DNA, that beautiful double helix, holds all the base pair instructions you will ever need. Yet not all of them are used, not every gene expressed. At any one moment, only a small fraction of your genome awakens to serve the body's needs. This is by design. It is how a skin cell stays a skin cell, though it holds the instructions to build a brain. It simply doesn't read those pages. Why? Because those pages are locked, marked, labeled by the soft touch of epigenetics, lock. Epigenetics tells your cells what genes to express, when, where, and how deeply. And it's done through incredibly subtle mechanisms, not by rewriting the DNA code, but by leaving chemical marks upon it. One of the most common is DNA methylation. A tiny methyl group attaches to a cytosine base, like a ghostly post-it note that reads, "Silence this gene." Another is histone modification dash, where small charges are added or removed from the proteins that cradle the DNA, telling it to tighten or relax, making the gene more or less available. It's like opening and closing shutters in the house of the cell, waxing crescent moon. And it doesn't stop there. Your environment, your emotions, even the air you breathe, can influence these epigenetic marks. Stress may shut certain genes down. Love may lift others into activity. A famine in your grandmother's childhood might still affect how your body processes sugar. Yes, some of these marks are passed down. Epigenetic inheritance. What your ancestors felt, what they feared, enjoyed, lost, can ripple gently into your own biology. You are not only made of genes, you are made of moments remembered across generations. Kale. That's why identical twins, born with the same DNA, can grow into people with different illnesses, different temperaments, different responses to the very same world. They began with the same script, but over time, each added footnotes, each underlined different chapters. Epigenetics writes experience into biology, not as fate, but as influence. Think of your brain. Each joy, each wound, each song you can't forget activates specific regions, and through repetition, those regions gain epimarks, neurons strengthen connections, proteins become more active, neurochemicals flourish or fade. That's experience carved invisibly onto molecular level. And when experiences are shared, in home, culture, or tenderness, these chemical echoes can bind generations together across blood and time. Baby. Now imagine a baby still in the womb, listening to her mother's breath and heartbeat, but also sharing the chemicals floating in her mother's body. If the mother is joyful, the developing baby may grow tuned to that calmness. If the mother is afraid, the stress hormones may leave notes on the developing DNA that say, "Be ready. The world might not be safe." These are not permanent truths. They do not doom or define, but they add complexity to the script, DNA. And here's the beauty in it all. Epigenetic changes are reversible. Unlike genetic mutations, these aren't hardwired. They change with care, time, choice. A troubled early environment can leave harsh markings, but new love, new learning, new safety can soften them. The cellular ink is smudgeable. Healing leaves footnotes of its own. So biology isn't destiny, not entirely. Even at the level of molecules, change is always possible. Rain. Scientists are now uncovering just how far epigenetics reaches, how it may influence aging, learning, risk of illness, response to trauma, how two people can eat the same food, but respond differently, based on chemical bookmarks left decades ago. These markers are everywhere, like breath on glass. We can't see them directly, but they shape the view. Suryan Marindu. What then does this mean for you? It means that your body remembers more than you realize, not just the things you say or do, but how you live, what you feel, and even what your ancestors felt. Your identity emerges not only from your genes, but from how those genes are tuned by life itself. It is not a rigid program, it's a living instrument, and epigenetics is the gentle hand, adjusting the strings, violin. So you are not just a product of biology, you are an interaction, a conversation between code and context. You are history, paused in flesh, then resumed in breath, in story, in choice. Bamboo. Tonight, as you rest, epigenetic changes continue quietly. A little more of this gene, a little less of that. Methyl groups rise and fall, like the tide inside your cells. You are still becoming, not just growing older, but shifting, always shifting, in response to the symphony of your life. Document. So yes, the script is written, but between the lines, you may still write your own emphasis, your own rhythm, your own chorus. Epigenetics doesn't rewrite the code. It teaches it to sing in harmony with you. Punje. And as we drift forward, we begin to wonder if these experiences are passed on. How far back does the memory go? And how does an egg, or a seed, store the echoes of generations? What does inheritance truly mean? You did not begin at birth. Your story started long before you opened your eyes, before you were held, before your name was ever spoken aloud. In a sense, you began the moment your parents met, or earlier, when your grandmother first carried the egg. That would become your mother, already holding a part of you. Yes, you were folding into the world, before you were ever a thought. DNA. Genes are more than instructions. They are gifts, whispers passed along through blood and breath and time, half from one parent, half from the other. A mosaic of traits shaped by choices no one living may remember. The curve of your ears, the odd way you hold a pen, the voice that echoes someone long gone. These are fragments not just of people, but of molecules. Silent, patient carriers of identity. Book. Within each of your cells, the DNA of your mother and father intertwine. In delicate reconciliation, two stories braided, retold as one. During conception, chromosomes align and exchange tiny pieces, a process called recombination. It's not mixing like paint, but shuffling like cards. A hand of genes is dealt to you, some familiar, some rare, some ancient. You are the only version of this story that has ever existed, or will. Punje. Some of these gene variants have wandered for centuries, from distant continents, from sunlit deserts, from frozen plains where life worked harder for warmth. Your resilience is global, carried in mitochondrial whispers and chromosome trails. These aren't just traits. They are survivor's marks, traits that made it through plagues, history's wars. You are walking evidence that uncountable generations chose to continue. World map. Inheritance is not just physical, it's also unseen. The smell of baking bread that warms you, the rhythm you tap your foot to, the grief you carry, without knowing why. It may not live in the base pairs dash dash, but it lives in how those genes are read, how cells respond to echoes of old joy, old loss. These are epigenetic memories, passed not as instructions, but as suggestions. Linger here. Sing more gently. Protect this child. They come from a long line of survival. You may look in the mirror and see a grandparent's eyes. Yes, because you inherited them. But also because eyes, like stories, follow bloodlines, not through design, but through repetition. Sometimes you are the first person born, with a certain smile. Since 1870, how quietly remarkable. Selfie? And not all stories arrive in pairs. Some genes are passed on dominantly, bold, expressive. One copy is enough to show itself. Others are recessive, shy, waiting patiently for a match across generations. That's how a trait can vanish for decades, then appear suddenly in one small child who looks like a painting no one remembers owning. That child is not strange. They are an echo. They are the chorus. Returning to the song, inheritance doesn't stop at traits. Sometimes it passes on broken parts, too. A missing enzyme, a fragile cell wall, a tendency for worry that lingers beneath calm. It is easy to think of these as flaws, but what is inherited is not fate, only possibility. Because for every gene passed down, there is a context dash, a world that chooses how it will be expressed. The same variant can mean trouble or triumph. Genes are a starting place, not an end. There is something humbling in realizing you carry cells with processes older than language. When you stretch, a protein loops through your muscles, first used by fish in prehistoric seas. When you cry, enzymes clear your vision, with molecules frogs once used. To see moonlight. Inheritance is not always about complexity. Often, it's about keeping what still works. Survival writes poetry through repetition, repetire, and yet, you inherit more than your genome. You inherit questions. Questions like, who did this first? Why does my voice feel familiar on tape? Why does loneliness bloom in me, even when I'm surrounded by others? These aren't always written in the code, but they linger in your tissues. They live inside reactions and reflections. Your ancestors faced their own storms and the shape of how they weathered them. Left fingerprints inside your physiology. Not just what they endured, but how. And still, you inherit the power to choose, to celebrate what you carry, but also to interrupt cycles. If a gene signals a higher risk for illness, you can change the environment, slow its expression, tip the equation. You are not your inheritance alone. You are where it turns into something else. Surya Udirvir. And maybe that's the deepest truth. That inheritance is only the first draft. Your life is the revision. With each breath, each friendship, each sorrow, you alter the rhythm of your cells. In what you eat, how you love when you sleep, you are influencing which pages are read and which remain folded shut. Bamboo. So tonight, as you drift, know that you are not alone in your body. You carry more than water and atoms. You carry people, places, decisions made in languages you don't speak. They are not burdens, they are lanterns, quiet shapes in the background of how your body becomes itself. Their stories move through you, not as ends, but as openings, magic wand. But what happens when we try to read this entire story? What happens when humanity turns its gaze inward and attempts to sequence itself, letter by letter? How close can we get to understanding the full text? And once we've read the book, can we begin to rewrite? There was a time when we searched the sky for meaning. We saw shapes in stars, prophecies in planets, signs in the sweep of comets. But one day, slowly, we turned inward, not just to emotions, not just thoughts, but to the deep molecular wilderness of our own cells. And there, in every nucleus, a code pulsed quietly, waiting. 3 billion letters written in the language of life itself, DNA. In 1990, a colossal vision began the Human Genome Project, a global effort to read, letter by letter, the entire DNA sequence of a single human being. Not just the familiar traits, eye color, height, blood type, but everything. All the proteins yet unnamed, all the switches yet unnoticed, all the silent sequences that may hold meaning down the line. It wasn't just about answers, it was about understanding the question. The numbers were staggering: more than 3 billion base pairs, about 20,000 to 25,000 genes, over a decade of work, billions of dollars, an international collaboration across continents and cultures. This wasn't just science, it was a quiet revolution, a world trying to step back and read its own code. For the very first time, document, imagine assembling a puzzle without the picture on the box. Now imagine that puzzle isn't static. It breathes, folds, duplicates, changes. Some sequences repeat endlessly, others remain stubbornly unique. Some seem to do nothing at all. Segments of DNA, long dismissed as junk, now revealed as deeply influential contributors to genetic regulation. What do we call the parts we don't understand? Are they meaningless? Or just silence we haven't learned to hear? Barker. As researchers mapped chromosome after chromosome, they discovered something beautiful and humbling. Much of our genome doesn't directly code for proteins. In fact, only about 1% to 2% of our DNA contains the actual blueprints for building. The rest? They manage the blueprints. They control timing. They moderate expression. They scaffold the structure and silence what must be silenced. It's not a code for building alone. It's a conductor's score, guiding tempo, volume, coordination. A symphony, not a schematic. And beyond that came wonder. We learned that 99.9% of all human DNA is identical. from one person to another. That means you and a stranger on the farthest continent, speaking another language, walking another path, share nearly your entire genome. It's in the 0.1% difference where diversity lives, your unique fingerprint of existence. We are more alike than we ever imagined, and more precisely different than we ever dared to know. World map. But the Human Genome Project was not just about knowing, it was about empowering. Suddenly, we could locate disease-causing mutations. We could trace genetic predispositions to cancer, to heart conditions, to neurological changes. We could ask, who is at risk? Who might need early screening? Who carries what? It opened the door to personalized medicine, the dream of tailoring treatment, not to the illness, but to the individual. And beyond medicine, a new realm opened: the study of ancestry, the molecular fingerprints of where we came from. People began sending in swabs of saliva and receiving the history of their blood. 10% West African, 40% Northern European. Traces of an ancestor from indigenous Americas. We had always asked, who am I? Now our cells answered, not with identity, but with origin. For all its promise, the Human Genome Project also raised questions, difficult ones, delicate ones. What does it mean to know our risks so early? Are we more human or more burdened by knowing how a gene might one day betray us? What happens to our freedom when our future is pre-printed in microscopic ink? Who owns that information? Where is the line between insight and intrusion? Lock. But the code does not command, it suggests. Genes do not determine your life. They open probabilities, possibilities, doors, not destinies. We hold the key. In environment, in choice, in epigenetic influence. You may carry a gene for anxiety, but whether it blooms depends on the weather of your life. You may have a variant for brilliance dash, but brilliance still asks for practice. There will always be more to a person than the sum of their sequences. Murray. When the project was declared complete in 2003, the sequence stood before us, astonishing and quiet. It wasn't the end of the journey, but the beginning of 1,000 new ones. Since then, the cost of genome sequencing has dropped from billions to mere hundreds of dollars. Now, anyone can sequence their code. Now, doctors can tailor therapy to mutation. Now, researchers can understand diseases down to a single misplaced base. The world shifted, not loudly, but like pages turning toward understanding. Still, even now, with every letter known, every chromosome mapped, your genome holds mysteries. Genes interact in ways we don't fully grasp. One gene silences another. One variant changes its meaning when paired with a different environment. We've read the book Dash, but we're still learning the language, book. And as we look ahead, past mapping into editing, we begin to ask, if we can read the genome, should we begin to rewrite it? Can we repair mistakes, erase disease, shape futures, Or is there risk in playing editor before we truly understand the story? Imagine this. You hold a page of music. Beautiful, ancient, familiar. But there's a mistake in the melody. A note strikes a chord that should not be played. And for generations, that off-key phrase has repeated itself. Now, for the first time in history, you can lift the pen, erase the discord, and write the note that should have been there all along. This is the promise and the poetry of CRISPR. Scribble CRISPR doesn't read the genetic code. It edits it, not with ink, but with molecules. Not on paper, but in living cells. It is one of the most profound tools ever discovered by science, and one of the most delicate. The full name is long, clustered regularly interspaced, short palindromic repeats. But the mechanism? It's elegant. CRISPR is the scalpel of the genome, a way to cut, remove, insert, or correct sequences in DNA with breathtaking precision. In nature, CRISPR is ancient, a bacterial immune system. When a virus attacks, some bacteria remember the invader's DNA and cut it up with molecular scissors called Cas enzymes, most famously Cas9. The bacteria keep genetic snapshots of enemies inside their own genome, like wanted posters. It's from this ancient microbial memory that scientists fashioned something new, a programmable tool, to target almost any gene in any organism. You write a guide, RNA dash, a molecular phrase that matches the target. You pair it with Cas9 dash, the cutting tool. Together, they find the sequence and change the script tool. This is not theoretical. CRISPR has already been used to correct genes that cause blindness, to delete mutations linked to inherited diseases, to edit rice so it grows more efficiently, to create mosquitoes that do not carry malaria. One simple, powerful, modular system, across species, across forms, across ethical frontiers. But with power comes pause. Because if we can fix genes, what else might we try to fix? Eye color, height, personality, and what happens when we attempt to define better? This is not science fiction. This is now DNA. There is a mythos that evolution is slow, a march across millennia. But CRISPR folds time. It lets us make in minutes what nature might take eons to refine. The boundaries we once accepted. The body can't do this. That disease will pass through bloodlines. This risk runs in the family. These are no longer constants, not because science wants to play God, but because parents want their children to suffer less, because gene therapies offer a form of mercy, where once there was none. Imagine a child with a rare condition, a single base error that causes their muscles to weaken, their lungs to pause. With CRISPR, we can target the faulty gene, correct the one-letter typo, and offer that child a life their ancestors couldn't have dreamed of. This is more than science. It is poetry written on bone, on breath, on future. And yet, not all edits are repair. Some peer into the realm of enhancement. Could we design humans to be stronger, faster, more intelligent? Could we ease the mind's burdens, make fear gentler, emotion more stable? Not must we, but could we? These are no longer philosophical questions. They are technical ones, and yet, beneath all the expertise, is a trembling truth. We don't yet fully understand the symphony we're trying to alter. Genes are not soloists. They are parts of complex ensembles, a harmony of influence. Change one, and the ripple spreads. Fix this, accidentally touch that. Our intentions may be pure, but nature's balance is older and more intricate than any one of us can grasp alone. So we approach it, not with conquest, but with quiet care. Many scientists call for caution. They seek strong ethical boundaries before human editing goes beyond repair into design. Others call for access, that this not become something only the wealthy can afford, a tool that could widen inequality at the level of the genome itself. This is more than science policy, it's a global reckoning, a philosophical return to an ancient question. What does it mean to be human? Magic wand. Perhaps in the end, genome editing invites not domination, but consciousness. It makes us more mindful, not just of what we are made of, but of what we may become. CRISPR lets us turn to our own cells, our own ancestors, our own mutations, and say gently, I remember you. But I must also try something new, something kinder, Barku. Even the name CRISPR feels poetic, like the sharp edge of autumn air, precise, clean, unfolding. It is at once a beginning, a blade, a breath. And if we listen closely, we might hear the genome singing back, "You may edit me, but do so as one who loves, not one who owns." Book. And maybe that's the truest path, not to perfect, but to participate, not to conquer code, but to understand its rhythm well enough to make a gentler verse. Tonight, as you sleep, your body repairs DNA quietly. Edits are made without your knowing. Small cuts, small fixes, guided by old repair enzymes that evolved long before humans even dreamed about themselves. Nature has been editing long before we learned the word. CRISPR, then, is not a rebellion. It is participation in a story already unfolding. Still, one question remains. If the genome is editable, is it possible that life too is editable in ways we never imagined? And if so, what do we lose when the mystery becomes manageable? What is the soul of the code we now hold in our hands? Before there were words, there were signals. Not spoken, not written, but felt. A molecule folded a certain way, and another responded. Message, reaction, meaning. No voice required. This, perhaps, was the first language. DNA. DNA speaks in 4 letters, A, T, C, G. A quiet script but deeply expressive. From combinations of these letters came the shape of skin, the color of eyes, the rhythm of heartbeats, and in time, it led to something wondrous. Not just the carriers of the code, but creatures who could understand that they carried it, who could tell stories about it, creatures like us. Humans did not invent language. We inherited the capacity for it, built on the architecture of the genome. Somewhere along the way, DNA encoded for muscles in the throat, wiring in the brain, timing in breath. From this biological scaffolding, sound became symbol, symbol became thought, and thought found its voice. Language was biology's next great experiment. The regions responsible for grammar and tone, Broca's and Wernicke's areas, are shaped by genes that no longer build muscle or pigment, but networks of connection. Our DNA began coding not just for what the body could do, but for how it could mean. Birdsong may signal territory, whale calls may mourn, but human language does something different. It reaches across time. We can tell stories of people we've never met, places we've never seen, and fears that haven't arrived yet. We can remember the future. We can imagine the past. Document. Genetic code gave rise to memory stored in protein. Spoken language gave rise to memory stored in story. Both are systems of continuity. Both preserve what would otherwise vanish. One in the cell, one in the voice. They mirror each other, and perhaps always have. Link. Think of how both codes are built from a small set of elements. DNA, 4 letters. Language, a handful of phonemes. Music, 12 notes. But it is not the number of elements that matters. It is their arrangement. A sentence tells the truth, or a lie, based on order. So does a gene. Swap one base in the BRCA1 gene, and cancer risk shifts. Swap one word in a vow, and a life changes. Both are systems of causality through arrangement. Book. Once, people believed that language arrived from the divine, a gift from the outside, a spark of soul. But if language is emergent evolution, then maybe the divine lived within the body all along, unlocked not by magic, but through the slow layering of change. Genes, brains, symbols, meaning. It is not hard to draw a line, from DNA to poetry. They both structure silence into something that moves us. Consider even this, the universal grammar inside all languages. Subjects, verbs, questions, negation. May itself be genetically coded. Linguist Noam Chomsky once suggested that language is an instinct. And now we are discovering that many aspects of speech light up in genes tied to developmental rhythm, to sound recognition, to the reward of meaning. Biology didn't just give us limbs, it gave us lyricism. And still, one must ask, why language at all? Why evolve the ability to whisper, to teach, to dare, and soothe and sing? Because connection has biological value. Early humans who could signal danger survived longer. Those who could tell where the water was, or recite the story of the moon belonged. Belonging preserved survival. The tribe's strength was not just in numbers, but in narrative. Parpugal. Even now, we hunger for stories. We seek them in strangers. We follow them in films. Our cells search for oxygen, but our hearts search for meaning. And perhaps that search is no different than the cells searching for a matching base pair or the strand seeking its complementary strand. Everything wants to fit, to be received, to be understood. So if DNA is a code, and language is a code, and music is a code, then maybe the universe itself is made to be read, not just through science, but through relationship. Each strand wants to resonate. You, after all, are a resonance of ancestors whose DNA remembered how to make a voice. Beep! And now here you are, listening to words shaped by molecules that built cells, that shaped brains, that wrote language, that carried thought into airwaves, into text, into your eyes. The code has come full circle. Parku. Tonight, as you drift into sleep, you carry a story inside your bones, not just of proteins and pigments, but of expression, of motion, of voice. The next time you speak, or read, or sing, know that it is not separate from science. It is science dash, softened into meaning. But gentle language isn't only a gift we inherit, it's also one we pass on. And deep within you, buried in the spiral of your own code, is a continuation still unfolding, still passed forward, into children, into generations, into what comes next. Beneath your skin, beneath thought, beneath the flicker of memory and the flicker of stars, there is something listening, something old, something patient. Every second, without asking, it continues. It copies, it corrects, it carries. You may not feel it, may not notice, but every moment you live is carried by the silent machinery within you. Your quiet program, DNA. The code, the ribbon, the archive. 3 billion letters curled gently in the dark, never demanding attention, never requiring applause. Only space and time. And you, inside every cell, the code rests, just microns wide, yet hundreds of generations deep. A seed, a scroll, a memory shaped into shape itself. You walk, you dream, you laugh, and beneath it all, your cells translate invisible instructions to repair that scratch, to grow in the dark, to make serotonin when sunlight touches your eyes. You are coded not just for being, but for sensing. Your sadness is molecular. So is joy. So is hunger and memory and even forgiveness. You are made of messages that speak back to the world. And yet, your DNA does not speak in words. It speaks in action. The protein kneels by the damaged strand. The enzyme clicks into place. The gene switches on or folds back to sleep. No orchestra, no fanfare, only rhythm, only grace. And in that grace, lies something sacred, not religious, but quiet, a kind of hum, a reverence, because this gentle code, unchosen by you, yet made to be you, has not stopped working since the moment of your first breath. It's a lullaby beneath breath, a pattern inside pulse, not loud, but true, parque. You are more than biology, but biology is part of your becoming. You are more than matter, but matter, lovingly arranged, became all your memories, your muscle, your whisper of longing. At some point, ribosomes inside your cells turned a sequence into a protein. That let you weep. At the look in someone else's eyes. This is both miracle and machinery, both science and story. Waxing crescent moon. They say you are stardust. That iron in your blood was first forged in an ancient star. That burst. But that stardust didn't know joy until it curled into code, built a brain, and remembered the name of a song. Your mother used to hum, "It's not enough to be made of stars." We are what happens when stars learn to heal. Galaxy. Still, the quiet program doesn't ask. It doesn't require gratitude. It continues, regardless of mood, season, or awareness. It is the steadiest companion you will ever have. Cells divide, genes express, epigenetic marks rise and fall, like tides beneath your consciousness. You are not just the passenger, you are also the ship, the sail, the storm, the home, house. The code does not define you completely. It opens a door, and you, by living, by loving, by faltering, step through it uniquely. The script is written, but how it's read is still up to you. Just as a piano holds every note, and yet only some are played with your hands. Piano, even now as you prepare for rest, the program continues. Mistakes made during the day are being repaired. Cells damaged by light or thoughtless impulse are cared for. Your DNA is not just code, it's commitment. To continue, to try again, to preserve what was offered from ancestors, and to quietly offer it forward as a freckle, as a talent, as a kindness, love letter. And when you dream, your body will still interpret messages, not in plot or dialogue, but in chemical fading, this gene dimming, that one glowing brighter. A choreography continuing, without witness, without applause. The program, loving, unseen, will write you into tomorrow. Document. So rest now. Let your awareness fall away, trusting that your body carries memory. More ancient than your name. You don't have to remember it all. Your cells do. Right now, a gene cradles a protein into place. A sequence is read by a ribosome like a lullaby. The system corrects, adjusts, forgives a mistake no one noticed. You are being loved by a million mechanisms below awareness. And every single one knows exactly how to take care of you. Nila, maybe the most beautiful code, is the one still being written, in kindness, in silence, in breath. And maybe The truest mystery was never out there at all. It was always within you, waiting quietly to be heard.